Yeah, thank you for that introduction. And I'm like wildly excited to be here. This is really great. Every conversation I've had has been really stimulating, and I hope I can return that favor with my talk. Very ambitiously entitled Multi-Scale Network Neuroscience. So the outline of the talk is I'll probably deviate from this a little bit, but just to give you kind of some structure. Give a kind of brief introduction to my field and the kind of work that we do, where our data comes from. I'm going to talk a little bit about what I call large-scale connectomics. I'm actually going to trash it a little bit. I'll start by trashing it, and then talk about much finer scale connectomics, single neuron resolution, and then remember all that trashing I did. I'm going to try to take it back and say nice things about large-scale connectomics using a single case study as an example. So I'll deviate a little bit from this outline, but that's broadly what I'd like to accomplish. And by the way, stop me if something comes up. I'm always happy to chat. I kind of like loathe the sound of my own voice. So anybody having a conversation that keeps me from talking makes things better and more interesting. Okay. So I think this is maybe not a surprise to people, but nervous systems are fundamentally complex networks. They are complex networks par excellence. There are special categories of complex networks in the sense that they are inherently multi-scale. And what I mean by that is that we can speak meaningfully about brain connectivity at different levels of spatial granularity. From the so-called micro or nanoscale, or considering the vertices and nodes to be individual neurons linked to one another synaptically, so very fine scale, single cell resolution. We can also meaningfully talk about connectomes, brain networks at a much coarser scale, where the nodes represent brain regions or brain areas that contain hundreds of thousands, millions, tens of millions of individual neurons, but they're linked to one another by large scale projections. So orders of magnitude coarser in terms of the resolution. Brain networks are also special in the sense that they're multimodal. The examples I'm giving you on the left, these are all what I'd call structural or anatomical brain networks. No surgeons in the room. Okay. I always ask because I occasionally give talks at like a medical school and there are surgeons. None of us are qualified to crack open skulls. Again, maybe you feel like you are, but you're probably not. Probably not. In principle, you could crack open a skull and you could touch these axonal projections, or you could visualize a synapse with a microscope. You actually see these things. You can touch them. They're physical. They're material networks that occupy space and volume. Right. So these are what I call like structural connectivity. But we can also consider another modality of connectivity, what I call functional connectivity. This is something that we get from recordings of brain activity. And the example that always shows up is the so-called resting state functional connectivity. This is from functional MRI. You record from a specific seed part of the brain. You get a time series. This is from the posterior cingulate. You can see this blue time course. You also record from the medial prefrontal cortex, another location in the brain. And when you compare those two time courses, lo and behold, they're temporally locked with one another. So we might be tempted to say that they are participating in the same kinds of functions. And so we've inherited this language when we say they're functionally connected at that point. I've been excoriated on social media occasionally by using that term, functional connectivity. Doesn't necessarily imply functional relatedness, but it's the term we have when we're talking about statistical relations between activity recorded from distant sites in the brain. That's the term we use. And here for functional MRI, which I'll talk a little bit about today, our measure of functional connectivity is usually a correlation. It's a very slow signal. Lags don't really mean very much for lots of reasons that I'll get into. But in principle, you could adopt virtually any measure of pairwise statistical dependence to measure functional connectivity. We still call it the same thing. Just have to remember we're measuring it in a slightly different way. And important to my work and lots of people in human imaging and non-invasive imaging, I like these last two cases, diffusion MRI and tractography and functional MRI, so I can acquire them non-invasively. I'll say more good things about kind of like large-scale imaging in a moment. So all of those examples I showed you in the previous slide, nanoscale to mesoscale to macroscale, functional to structural, they're capturing some kind of pairwise relationship. So we need a framework that's really well suited for representing pairwise relationships. And so enter the network model. I don't have to say a lot here about this model. It's kind of a stock slide. But obviously the network, our things become the nodes, the items in our network become the nodes, and those pairwise links, structural or functional connectivity, become the edges. Network science is having a day, and it has been for 20 or so years, right? I can't think at this point of a scientific discipline that it hasn't infiltrated in some way, from microbiology to sociology and anthropology to economics. Even the environments that we're embedded in have structure to them and can be meaningfully modeled as a network. So it's everywhere, right, including neuroscience. And we like this. It's a common modeling framework, abstracts away a lot of details, but we're left with this circles and lines representation of the system we care about. We can share tools across discipline, which has been really profitable for us network neuroscientists, especially in the early days. And to the extent that you care about it, we can consider networks as a special class of system unto themselves, interested in trying to understand maybe universal patterns, features that are shared across networks of all types, or at least real-world networks. So a brief word on how we construct macro-scale brain networks. Anybody had an MRI scan before? A couple hands going up. Functional MRI, structural MRI, or for muscles. Sometimes all of the above. Sorry. It is a lot of fun. So it's like very polarizing. Either people like really hate it, or they like, some people like, it's womb-like being in the magnet. It's very comforting. It's very, other people like, no, it's loud, it's cold, it's hot. It's very loud. That's one that people complain about a lot. I'll tell you like an anecdote a little bit later about what prolonged exposure can do to your ears. Don't go to the magnet every day for like a year. It's basically the upshot. When we build brain networks, we really consider two flavors of MRI. One called diffusion MRI, which measures the apparent motion of water molecules. When those water molecules are bound by fatty fiber tracts in the brain, they diffuse anisotropically. And so we can indirectly infer the presence of white matter from diffusion MRI. And again, this is all non-invasive. It's a $500 scan. You can do it for any one of you, as long as you don't have dental implants or like ferromagnetic things in your body. We can get this kind of data from you. We can also measure bold activity, which is a hemodynamic response. So your blood is to population level neural activity. When a bunch of neurons become active, it's metabolically expensive. They need oxygen to continue being active, and so the blood will be redirected to those areas. And we can measure that indirectly and non-invasively. So it's not a direct signal, it's not neuronal activity, but it's this kind of secondary measurement of the hemodynamic response to that activity. Once we have these kind of data, these inferred fiber tracts, the bold activity from different parts of the brain, we typically chop up our brain, again, not literally, but figuratively speaking, into parcels. Those parcels become our nodes. We link them if the bold activity is correlated or if there's a streamline between them. From this, we get our structural connectivity and our functional connectivity. The next slide, I'll say a few more words about this. They're very different in character. They capture very different features of the system. But from those matrices, we can do all of our fun network analyses. Right, I don't think I need to say a ton about that here. So I also know I'm not speaking to neuroscientists. Is this part kind of clear? Although with some rare exceptions, but like this is the basis for where we're going next, I think. Okay. So we get these two coupling modalities, structural and functional connectivity. I'll show you some examples. These are real data now. These are from human MRI. Structural connectivity on the left, functional connectivity on the right. I love doing this. I'm like a very visually oriented person, so I like seeing things. I'm just going to toggle back and forth between the two, hoping you can kind of see they're not identical, but they're still interrelated in some way. There's definitely some shared correspondence. And indeed, they are similar in lots of ways. We can represent both structural and functional connectivity as networks. We might say they have multi-scale organization themselves, meaning we can speak meaningfully about fine-scale features of these networks, properties of edges and nodes, as well as properties of the network as a whole. But they're also very different, as I said before. Structural connectivity is those physical connections. Structural connectivity is sparse, meaning most connections that could exist don't exist. In principle, over a short enough time window, Structural connectivity is also finite. We don't grow new brain regions. We don't grow new fiber tracts. Those fiber tracts, they have complex biophysical attributes related to the density of axons, the conduction velocities along these projections. And they do change, but they change very slowly. They change with experience, with plasticity, and they change over programmatic development, aging, and ultimately into senescence. Right? They're constantly fluctuating, but on a very slow time scale. And the way we think of structural connectivity, it is this physical backbone. It is material connections. It's kind of a constraint on which parts of the brain can talk to one another. So it's shaping and sculpting patterns of communication between different parts of the brain. Functional connectivity, on the other hand, as I said before, we usually estimate it from something like a correlation. So it is statistical and therefore dynamic in nature. It is capable of changing over very short time scales. Functional connections have their own potentially interesting attributes, their persistence over time, their directionality. But functional connectivity is also context dependent. And what I mean by that is if I put you in the magnet and ask you to do a cognitively demanding task, we might expect your functional connectivity pattern to change. It might change if we were to zap your brain with a non-invasive stimulation, like transcranial magnetic stimulation. We might expect that to in some way alter your pattern of functional connectivity. And the way to think about it just overall is kind of the outcome of the communication processes happening. Communication is constrained by the wires. As a consequence of that, we can measure the correlation structure of those constant communication events that are happening in the background. And this is kind of like, in a nutshell, the bread and butter of network neuroscience now, especially large-scale network neuroscience. Just a quick word on network neuroscience as a discipline. I think historically, it gets reduced occasionally to like network neuroscientists. Oh, you must be doing graph theory. It's like they say this, right? If you talk to neuroscientists. I think that was true maybe 15 years ago when people like Olaf Sporns first kind of pushed this idea of representing biological neural networks as graphs and doing fun stuff with that. But it is now, I think it's a mature field. It has its own sub-disciplines, even at the large scale. People are very interested in using brain connectivity to constrain biophysical models. So you imagine like smaller subpopulations of excitatory and inhibitory neurons positioned at each location in the brain. They're governed by biophysics, and we can actually simulate these in time, coupled to one another by the wires, and it generates synthetic data that kind of looks like the real deal, and it allows us in silico then to test predictions about what would happen if somebody had a lesion that destroyed part X of the brain. Well, we can make a prediction in silico using biophysical models. There's other more abstract models of communication between brain areas. People are very interested. This came up in some of the discussions the other day about how brain networks change over very short time scales. There's a lot of interest in trying to understand the principles by which brain networks reconfigure moment to moment. We and others have done a lot of work building generative models of networks, trying to understand a low-dimensional set of principles that when you encode them explicitly into wiring rules, generate synthetic networks that capture features that are also observed in real-world brain networks. There's a lot of work showing some controllability papers, but there's interest in trying to understand how we might deliberately modify the network by stimulating it to drive it into particular configurations. And I'll say a lot more about this at the end, but one of the areas I think is most exciting within this field is so-called precision mapping. I'm not going to say a lot about it now, but it's kind of in the same spirit as precision medicine. Historically, network neuroscience and non-invasive imaging kind of warp everybody's brain to look alike. We force everybody to be more similar to one another than they are. But over the last decade or so, it's been appreciated this is probably not a good idea if we want to make real advances, and that our brains are very unique and stably so. And I'll show you some examples of how we can leverage that in a clinical setting. That'll be the final chunk of this talk. And people are also interested in other things like low-dimensional embeddings of brain networks. So it's a very active field, lots of different areas of work. It's very still kind of a fun time to be a network neuroscientist, I think. So again, I'm going to pause. Are any questions? Okay. So I've told you a little bit about the field. I've told you a little bit about where kind of brain networks I usually use in my own work, where they come from and how we build them. So I want to talk a little bit about like large scale connectomics and my lab's involvement in that. So I think a lot of the work that I do, it starts with this, it's like really a singular question. When I get a connectome, when I get a brain network, why the hell does it look the way it does? Why are particular parts of the brain connected, and why are particular parts not connected? And historically, we've kind of approached this from, it's maybe the wrong terminology, but we're trying to understand, I guess, like organizing principles in some way. What are the key dimensions of brain networks structurally? Kind of like a list of parts. If I had to say, like, these are the core elements that every brain network must have, what might they be? And beyond just giving you a list of parts, which isn't particularly constructive, I'd also like to know what that part is doing in terms of brain function. What is its role individually, but also when we put them together, how do they interact? How do these parts list come together to support normative brain function? Yes, quick question.
These are the things from the neurons that go close to another neuron, right? And then they release substances.
Yeah. So wait. So I'm sorry, I'm misunderstanding. Which? Yeah. So
arms that from neurons
that's an example of a connectome, and the pending "ome" to the end of that, I think, implies completeness. So it's not just a little chunk of tissue with all of the wiring, the neuron to neuron synaptic connections, or even zoomed out, like big fiber tracts between different parts of the brain. It is somehow the entirety of the organ. It's the wiring for the entire brain. So when I say a connectome, I'm trying to imply that it's an entire organ. It's the wiring for an entire organ. And it could be at different spatial scales, like you said, from individual neurons all the way up to a much coarser representation.
Also collective phenomena.
I wouldn't call it collective phenomenon in the sense that it's just a different tool for measuring your data. If I had really good microscopes and good machine learning, I could get those single cells. But if I put somebody in a magnet, like an fMRI, I do an MRI, I could also build a connectome. I can never see in the magnet those single cells, but I can still meaningfully connect two parts of the brain on the basis of a different kind of connection, a big, thick, fatty wire between them. Yeah, of course. And for better or worse, I think especially the large scale network neuroscience world is kind of converging onto a set of like conserved, key, almost canonical organizing principles. We and others have talked about this at length. These include brains are modular, right? They exhibit assortative modules. We think that these help support specialized brain function. It's different parts of the brain that are densely interconnected internally, but weakly connected to one another. Brain networks exhibit heterogeneous degree distributions, so to the point that there's a small number of highly connected nodes. We see this both at the single neuron resolution, but also at the large scale. And that gives those highly connected neurons, those highly connected brain regions, greater capacity for influence. More than that, those highly connected regions connect to one another, forming something called a rich club, this highly integrative structure. Brain networks also exhibit efficient processing paths. And what I mean by that is they tend to have short path length on average. So it's very easy to send a signal from one part of the brain to another in very few hops. It's something we think is related to the need to very quickly send information from one locale to another. There are motifs that support specific brain functions. And possibly above all, brain networks, especially the connectome, those physical wires, they're embedded in three-dimensional space. Forming connections requires materials, it requires energy to maintain them, and it all has to be encapsulated within a skull or an exoskeleton. There's limited resources. And so brains have, over evolutionary time, seem to have adopted an economically efficient wiring diagram. They favor short-range connections because they can put them in a skull. We can put many of them. They favor short-range connections because those are less costly than a very long tract. In my lab, in trying to address these questions, I think we focus largely on the macro scale, the human, non-invasively acquired brain networks. There's a lot of reasons to like this. There's a ton of reasons to stay at the macro scale, right? As I said before, MRI scans, not that expensive. There's a big indirect cost of like having a magnet and filling it up with liquid helium every so often, having a guy from Siemens on hand when it breaks to come in and fix it, or from GE to come fix it. It's like $500 a pop. That's not bad, like experimentally. It sounds like a lot. Like I'd love to have $500 in my wallet right now, but like it's coming from my grant budget, from startup budget. It's not that bad, right? Compared to other imaging techniques, functional and diffusion MRI give us whole organ coverage, right? If you come from a systems neuroscience lab, you study visual processing or auditory processing or memory, you zoom in on a select set of areas or a select area even. You image those neurons alone. You don't know what's happening in the rest of the brain. Functional MRI for all of its problems, diffusion MRI for all of its problems, it gives us the entire organ. It gives a picture of the complete system, which is really, we can leverage to our advantage. As I said before, it's non-invasive. If you work with mice, if you work with monkeys, it's term limited for those mammals. Sorry, like at some point the experiment ends and you're obliged to humanely sacrifice. I don't like doing that with my human participants. I think there's big ethical issues with that. So it's non-invasive. MRI gives us the ability to non-invasively take inexpensive whole organ pictures. It is applicable to mammalian brains. I have lots of colleagues who do non-human primate imaging, who do small mammal imaging, which includes rodents. You can do this. And in fact, you can put them in really, really high magnetic fields to take really exquisite pictures of their brains. But just a note on the field, I think this is true. It's not true of every scientific discipline, but people working in non-invasive large-scale imaging, we have like the best data sharing ecosystem I can possibly think of. Not only does NIH require us to, like, there's a mandate for sharing data, we also have standardized data formats. We have repositories that aggregate across studies to the point now, if you want MRI data, you press a button and you download terabytes of somebody else's data that's already well curated, possibly post-processed, and you're ready to go. It's a very, very nice ecosystem to work in. As I said before, my work in this space historically has touched on lots of topics, from generative modeling to trying to understand what the role of these costly long-distance connections are. I said brains are economically wired. They have long-distance, costly connections. They must be doing something special because they're so damn costly. Why would they be there otherwise? So we've done a lot of work trying to understand that. We've crossed modalities, still using human data. We've worked closely with neurosurgeons who implant electrodes directly on brains. So we've kind of cross-validated our non-invasive imaging with direct invasive imaging of individuals. We're very interested in the notion of dynamics, trying to understand how and why brains reconfigure over time. And we even touched on this kind of current hot topic of higher order connectivity and higher order organization in some recent papers. So we touch it all, like, in our quest for understanding organizing principles. This is, like I said, I'm going to say some bad things about MRI. This is where I start. This is like where I like cut my own legs out from underneath me. Things I've said already. MRI is indirect. There's no gold standard to compare against. We don't have when we measure an MRI, we get back what we get back. Registering it with something real, something like invasive, is really challenging to do. We don't have a benchmark, right? As an example, like I said, with diffusion imaging data, it's based on the apparent diffusion of water molecules. We make this measurement at each location in the brain. We fit a model for the preferred direction of water motion. And we build plots like this, but there's no sense of, it's never registered against invasive techniques, because you can't do this with humans. There's lots of sources of error, some of them really damning, in fact. One of the big challenges with diffusion imaging is the so-called crossing fibers problem. If I have a voxel and I have lots of fibers at many different orientations entering that voxel, my model might say, actually, because they're all at different orientations, diffusion there is isotropic. There is no preferred direction because they're all intermingled, or it forces that voxel to have a single preferential direction of motion. So I miss out on a bunch of connections that actually should be there. There's other insidious issues too, like in-scanner movement. We ask people not to move, but invariably they do. We make people wear customized face guards that are locked into the, yeah, and we still move, everybody does. And this is now a pain study, by the way, when you do that, because it's like really uncomfortable. Some people sweat, some people have beards, and like, you don't want to be in like maybe you do, maybe you actually want to be. I don't want to be in a mask like that for while I'm in the magnet. MRI also has really limited spatial resolution. The finest you can get, about a millimeter cubed. That millimeter has like 100,000 neurons in it. You're blind to that. You can never see that with MRI. You can't go finer than that scale. And consequently, it's very difficult to link to neurobiology. So, again, as much as I love the non-invasive side of my work, linking it to the neurobiology is very challenging. And because we work with humans usually, it kind of limits the kind of interventions we can do. Right? So there's all of these issues with issues. There's these challenges of doing large-scale network neuroscience. So I've trashed my own field. I'm sorry, everybody. In case there's any network neuroscientists listening. Okay. So a quick personal anecdote. I started my lab at IU in 2018. Turns out it was a really bad time to start a lab because a couple of years later, I didn't know it at the time, but we're going to be shut down and never see my students. But at that time and just before it, I kind of made a promise to myself. I had done mostly large scale network neuroscience at that point. It was kind of dissatisfying for all the reasons that I said before. It limits the kind of statements we can make. The non-invasive nature, the indirect measurements, you can only say so much when those are your data. So I had this plan of gradually shifting away from non-invasive large-scale network neuroscience. And it's work that we actually started as a postdoc. I started kind of trying to do this as a postdoc, including actually running experiments in labs. I collaborated with people who do systems neuroscience, doing 2-photon imaging of mouse auditory cortex. I hated it, but wrote some papers. Nice, generous labs, made really cool data available. So we started messing with that. This is zebrafish light sheet imaging data. It's an entire brain, 80,000 neurons, really slow, but it's still really nice. And it gave us a chance to do something that wasn't human, which was exciting. And of course, we worked with all of the non-human mesoscale connectomes that were out there. This is mouse, macaque, rat. They're out there, but they're still at a very coarse resolution. Right? They're not single cell, single neuron. And so that was kind of dissatisfying. And because we had some nice grants early on, got lucky, kind of put that ambition on hold for a little bit. But then in the last few years, because really massive advances happened in network neuroscience. They reconstructed at single neuron resolution, single synapse resolution, connectomes. So the wiring, the physical wires, for an adult and larva Drosophila. It's who cares about flies? But it's a whole brain. It's at single resolution. And they actually have a ton of shared data. There's a lot of overlap in terms of genes. 60% of our genome is the same as a fly. Something like 3 out of 4 human brain disorders. There's an analogous disorder in flies. There's lots of reasons to like this. But for me, somebody who wants to understand how connectomes are organized and why, this is one of the first times where we have a complex organism, worms notwithstanding, but a complex organism with a complete wiring diagram. So this is a really exciting time for me. So the data come from electron microscopy. Entire volume has to be segmented, proofread, annotated. I'm thankful I'm not involved in this because it seems like a really painful process. It's a multi-institution, multinational collaboration that ended up generating these data sets. But in the end, you get back things like this. So on the left, this is the larval Drosophila. This thing dangling down here is the ventral nerve cord. And over on the right is the adult. 140K neurons in the adult, 3,000-ish neurons in the larva, okay? And this is where it gets cool. I mean, it's already really cool. But where it also gets doubly cool is that both of these data sets, they come with tons of rich neuronal annotations. These are not annotations that necessarily come from like a machine learning algorithm. These come from expert neuroscientists. We're going neuron by neuron, attaching manual labels to these. So I know that seems arduous and probably is, but I think it's nice as somebody who wants that knowledge in their own work and can't do it themselves. Like I could never hand label neurons in any meaningful way. I could try, but it'd be really bad. This is just one example, a very small sliver of the annotations that accompany these data. So it's very exciting to us. It's a way to take our connectivity data and explicitly link it to brain function, to annotations, to cell types, to you name it, right? And so finally getting to data. How am I doing on time, by the way? Don't tell me I have 15 minutes. We're good? Okay. So I want to talk about two studies, one published and one still in the works. Our first paper came out in PNAS a year ago with co-authors Kyle Seguin and Maria Grazia Pochedo, who's actually now a faculty member at Virginia Tech. She just got hired. Again, bad time, I think, to be hired, but at least she has a big pot of startup money. It's like NIH says, we're not funding things anymore. But yeah, so two really great scientists. And then the last, or rather the second paper is analyzing the adult connectome. A lot of overlap in terms of the research team with another close collaborator, Bratislav Misek at the MNI. Just as an aside, especially to like young scientists, it's like so much fun to do science with people you like. And like I really like these people, so it's like a really great. It's just fun to do science. Like yeah, it's fun to do science with people you like. Suffice to say. Okay. So in series, I'll just go through what I think are some salient points from these papers. So just like our large-scale connectome data, we have wiring diagrams for these organisms, synapses linking individual neurons. We also have annotational data, which is kind of new. And so the question for us, does network structure, does the organization of the connectivity alone, does it carve nature at its joints? And what I mean by that is, are there features of the wiring that in some way respect these annotations? Because they don't have to, okay? This is something that we couldn't test very easily at the large scale, right? We have annotations, but there are also other MRI modalities. We have like PET imaging to tell us about the metabolic activity of a region, but it's still not invasive. Here are expert annotations with near ground truth connectivity. So our focus was on community structure, something we have done a lot of work with. I was bullied online by somebody, not going to name who, to use their tools, but it's actually a really great tool. I've come around to really liking GraphTool. I started like years ago during my PhD, like nobody could install it in the lab. It was like this like long-standing joke. Finally, somebody shared binaries and like easy Conda installs. So now we have it. It's really great. I joke. I'm very happy to have used this and continue to use it. But we're using a weighted, nested stochastic block model to detect hierarchical communities. You can kind of see what those co-assignment probabilities look like. These are neuron by neuron. There's the hierarchical levels, and we're going to focus on a specific level, kind of an intermediate scale. Why an intermediate? Sorry, this is larva. This is the larval connectome. We're going to get to the mother Drosophila in a moment. This is still the baby. Yeah. So we have the communities. I'm going to focus on a kind of an intermediate hierarchical level. Why? Because it's not super coarse and it's not super fine. Kind of balances the size of communities. And these are in anatomical space what some of those communities look like. It may not mean anything obvious if you're not a fly biologist. Some obvious things that I'll point out. Neurons in the same community are colored the same, so they co-localize spatially, which is really nice. It's a good sign. It's not like a random assortment. But the question then is, do they map on to these annotations? Are they related to these labels for cells? And so to do this, we calculate what we call an enrichment score. We did this for every pair of annotations and every pair of communities. Okay. How did we do that? Took our annotation. So this is a label for a set of functionally related neurons associated with mechanosensation. We took our community labels and calculated a measure of overlap. Big things and small things that will kind of bias our overlap score. So you compare the observed overlap against a null distribution. It's a very special kind of null distribution. It's not a random permutation of the labels, except fails to preserve spatial relationships. We use a tool called, you ready for this, brain smash, but it takes a set of three-dimensional points with a label. It measures something called the spatial variogram, which is a kind of spatial autocorrelation in your data. And then you can fit it to your data to match your data's variogram. And then it generates synthetic data sets that have approximately the same spatial autocorrelation, but is otherwise randomized. The actual labels are random. So we're preserving space, which is an important constraint on brain networks, but we're otherwise shuffling the labels. And then we repeat this process, calculate another measure of overlap, do this 10,000 times. And we build up null distributions. And so then we express our enrichment scores is our observed overlap, z-scored with respect to that null distribution. So big z-scores imply that there's greater overlap than we would expect given the spatial autocorrelation in our system, given the size of the annotation and community. It's kind of like how overexpressed is an annotation within a community. And I'm showing you this here for just this one sample. And so there's some possibilities for the outcome, right? It could be the case that community boundaries don't matter. You kind of see a smooth and graded response in these enrichment scores, right? Or it could be the case that specific annotations are almost entirely enriched within a community and then nowhere else. So are communities enriched for specific annotations? This is a subjective statement, but to me, the answer is seemingly yes. So I'll give you an example of what I mean. Consider community 6. So these are functional labels, functional annotations related to gut function, respiration, and gustation. Community 6, it's those labels, those annotations, and only those annotations. Everything else is basically at baseline. There is no intermingling, right? There's almost no graded relationship to any of the other annotations. Another example is community 2. It's enriched actually for nothing in terms of functional annotations, but it's deeply enriched for something called mushroom body input and output neurons. And the same is true for basically every community that we detect. This is true also for the other scales of the hierarchy. It's more or less the same, which is kind of exciting. The way I think about this, I'm not making a claim about directionality or causality, but the connectome wiring and these cellular annotations are kind of flip sides of the same coin. I don't know which causes which. I don't know if it could be the case that certain cell types preferentially connect a certain way, or it could be the case that connectivity kind of helps shape the functions of these neurons. I don't know which is the direction that goes. But that was really interesting to us. And so actually from that second paper, because that first PNAS paper was just the larval connectome, we asked, do we see the same thing in the adult? Now the adult is much bigger, 2 orders of magnitude more neurons, the annotations are much richer, and the communities consequently, we find many more of them, even with the same hierarchical blocks modeling approach. But I'm showing you an analogous plot. Each sub-panel, those are different flavors of annotations. And again, I draw your attention to the fact that consider just the class annotations there. So every neuron is associated with a very specific class. We have, in this case, more than 1,000 communities at the finest scale. It's not a very graded relationship at all, right? It's a nearly all or nothing. You're enriched for something or not. There's not a lot of intermediate colors. I wish I could say that this was due entirely to my choice of color map and scale, but it's not. You kind of, no matter how you slice this, you observe this kind of behavior. So it seems to be true even in the adult connectome, which is, again, 2 orders of magnitude bigger, more complex. Adult flies can do things that in terms of their behavioral repertoire that the larva cannot do. And again, just to show this to you visually so that you can, this is not just some like statistical fluke. These are Kenyan cells, which are cells associated with associative learning and olfaction. And these are a handful of communities that have high overlap with those cells. These come from just connectivity alone. They don't know anything about the annotations. But boy, they line up really nicely with our Kenyan cell labels, right? Which is kind of remarkable. Another example, these are olfactory sensory neurons. And here's another community that has striking overlap with them. So it's not just a figment of statistics. You can see it visually too, which is, I know that's not how we should be doing science, but it's very convincing to me when I can see the effect I'm describing. So going back to the larval connectome, so we started asking, well, communities are nice, but if you read that large scale imaging literature, we talk about other kind of core organizing features, right? Ridge clubs, for instance, I talked about that before, the highly connected neurons that are also connected to one another. How are those aligned with these annotations? So first, we do indeed find a ridge club, find many ridge clubs where they're statistically significant, but we're going to focus on this peak. Here's the composition of the ridge club. These are where the rich edges fall. The connections among ridge club members are overexpressed, which is what they should be. And I'll note that our ridge club members, those are those communities that didn't have any functional annotations, incidentally. It was kind of interesting. And sure enough, our ridge club tends to be highly connected interneurons, including things like those Kenyan cells, which there are progenitors in the larval connectome, as well as some more of those mushroom bodies. So there seems to be some enrichment for the ridge club as well, right? We can even flip this enrichment analysis. So rather than asking, are network features enriched for annotations, we can ask whether certain annotations are enriched for network properties. Again, we really like community structure in my group. And one of the things that we can do when we have a block model is we can characterize how different communities interact. And there's different motifs that they can exhibit. For any pair of communities, they can interact assortatively, so it's internally dense, externally sparse. They can interact as a core and a periphery or as a disassortative motif. And so we can ask, well, are our annotations enriched for these different labels, these different network labels? The first point I'll draw your attention to, almost all the communities are assortative. Even though the block model is in principle sensitive to these other interaction types, almost all of this segregated sort. So the modules tend to be segregated. There's a small subset that aren't. Showing you some of these anatomically. So here's our assortative modules. They involve neurons that have projections down to the to the ventral nerve cords, so associated with like the rest of the body and proprioception. But we also have some non-assortative neurons. These are the ones that form cores and peripheries. And those, again, as it turns out, are enriched for interneurons. But rather, the interneurons are enriched for this kind of not higher order, but non-assortative motifs. Which is kind of exciting for us. Okay, so that was there's more in the paper. I'll let you read it if it's interesting, and happy to chat about it. I want to talk about one other study, the second one that's still under review. If any of you are reviewing it, boy, I'll buy you a drink or something. We like this one too. If you don't drink, I'll buy you a sandwich. I don't know. So the last paper was mostly looking at community structure and linking it to annotations. We asked ourselves, well, are there other ways that we can leverage these fine scale data sets with rich annotations to better understand a brain function? We talked to lifelong network neuroscientists like Olaf Sporns or Danny Bassett. They're going to tell you that one of the things that every connectome is, it's a communication network. The wires allow different parts of the brain to talk to one another. That is one of its chief rules. And among the information that needs to be communicated from one part of the brain to the other is information from the environment, information that's transduced by sensory neurons and relayed to other parts of the brain that are associated with, say, navigation, decision-making, or action planning, okay? So what we wanted to try and understand, using the adult connectome, how do these sensory cascades unfold, and what is the connectome's role in shaping them? Am I over time? Okay.
There's a class also.
I'm noticing people filing out. That's why I was a little bit nervous. Yeah. So I'll go through this quickly because I really want to touch on the last study too. But essentially, we're going to model sensory cascades using a subset of neurons that already have sensory annotations for different sensory modalities. These are going to be our inputs. And we're going to use a really simple model of synaptic activity, of transmission of information. It's a three-state model. Neurons can be active, inactive, or quiescent. They're going to probabilistically activate their postsynaptic partners. It's literally a Bernoulli trial with probability P, and we're going to flip a number of coins equal to the number of synapses. Okay? A single success, as we're defining it, is sufficient for a presynaptic neuron to activate its postsynaptic partner. And then once a neuron's active, in the subsequent step, it becomes inactive. So it's a discrete time model. It's very simple to implement, but it's also probabilistic and stochastic. Right? We're going to look at three flavors. I'm trying to go through this quickly because I really want to get to the final study. But we're going to consider what I'll call unimodal cascades, where we seed our sensory neurons and allow the cascade to propagate until completion. A cooperative cascade, where we seed different sensory modalities simultaneously. So it'd be like a polysensory or multisensory integration. You have an auditory stimulus along with a visual stimulus, as opposed to just a single unimodal set of sensory neurons. And then we're going to consider a multimodal stimulation case where they're going to interact competitively. There's like two different seeds, two different families of signals propagating over the network, competing for territory. This is what a typical cascade might look like. Actually, this is a summary of many cascades. It's telling us the probabilistic timing of activation, so the likelihood that a neuron at a given step is active. These are what the probabilistic maps look like as they unfold over time in anatomical space. In this case, we're seeding all sensory neurons to do this. This allows us to address a whole bunch of questions, like how similar are these trajectories from one sensory modality to another? Upshot is early on, they're very dissimilar, but gradually converge on a set of shared targets. We can ask which pairs of sensory modalities should we stimulate simultaneously to reduce the total amount of time, to reduce the activation time by the greatest amount. It turns out because the visual neurons are, in terms of their connectivity, they're very distant from the rest of the network. If you pair them with anything, you tend to get a big reduction. And we can even ask questions about where convergence happens in the brain. We can literally looking for theoretically motivated sites for multimodal integration. And we do this in two different ways. Well, I'm going to run through this really quick because I really want to get to the last part. The upshot is we can do this, and we can make predictions that a neuroscientist could, in principle, go back and validate experimentally. Sorry, I'm running through this quickly. Okay. So I said I'd say bad things about large-scale imaging. I did that. I said I'd tell you about a couple studies at the nanoscale. And now I want to go back, right? So nanoscale connectomics, awesome. There's really great spatial resolution. We see things that a large-scale imager just plain old can't see. The annotations are useful for forming a bridge to neurobiology. And in principle, if you were so inclined, you could coarse grain your data. You could start small and build up to something much with less granular in some way. But there's still an appeal of large-scale imaging to me. It keeps drawing me back in. Those nanoscale data sets I showed you, one sample each, one brain each, one animal each. Now, there's a lot more stereotypy in flies than there are humans. So we might expect one fly to look a little bit more like another fly and one human brain to look more like a human brain, but it's still a single sample. It is expensive and time consuming to generate those types of data. And as somebody who used to work in a psychology department, works in a medical school, and works in a neuroscience group, institute that cares about the developing child brain, those kind of studies, as cool as they are, they actually don't inform our day-to-day practice very much. It helps us understand these kind of generic wiring principles, but it doesn't give us a lot of insight into what we can do to help humans. So I'm going to talk now, this last bit, about how we might salvage this, how we could salvage large-scale imaging so that it's still useful. I said a lot of bad things about it, but what can we do to make this better? So the first point, I'm going to point to two big limitations I haven't discussed yet, right, but I think that we can overcome now. Translation is a big key in working with humans. We want to do experiments. We can translate to to medicine, to treatments, to something that helps people. But if you look at the literature, literally no fMRI studies translate. Nobody is using fMRI clinically. There's a handful of people who use it for like localization of like motor areas pre-surgery, like pre-surgical planning, but it's not really showing up as like it's a research tool. It's not a clinical tool really. Part of the reason it doesn't work, because the kinds of effects that we see in large-scale brain networks, they are teeny tiny. But people do something that's called a BWAS, brain-wide association study, borrowing that term kind of from GWAS. The effects that you see when you have massive samples are really, really small. And to measure those effects reliably, you need huge consortia studies of 10,000 brains. This is an example of an association of brain connections with a measure of intelligence in 3 massive data sets: the Human Connectome Project, the UK Biobank, and the Adolescent Brain Cognitive Development Study. The range of correlations is super narrow. The effects are super tiny. They're very difficult to measure. To get stable estimates, you need thousands and thousands of brains. Otherwise, your estimates are, it's all nonsense. There's a lot of error. The other issue is that when we do analyses of human brains, we force brains to look alike. It turns out not everybody knows this. I was really surprised to hear that yesterday. So that we can do statistics and compare a bunch of brains, we take my brain and warp it to a template. Take Brendan's brain, warp it to a template. We make everybody's brain look artificially more similar than they are. We're washing away tons of the things that make my brain special that we might actually want to target clinically and as clinical practitioners. You've washed that away. A great example comes from a study of Russ Poldrack. This is where like the scanning yourself many times comes in. So Russ volunteered to scan himself 3 times a week for more than a year. He generated hundreds of scans of himself. And what I'm showing you here, this is a community structure for a group brain and the community structure for Russ's brain. And I want to point out this looks like noise to some people. They're like, oh yeah, it's not as smooth and it's not as consistent as the group. But no, this is still thousands and thousands, not thousands, but hundreds and hundreds of minutes worth of data, hours and hours. And it's reliable, these little blobs. They're very Russ-centric. They're reproducible within his brain. But if we were to treat his brain identical to other people's, we would wash out all that variation. This was further made even clearer from the Midnight Scan Club. This is a group of researchers at WashU. It turns out scanning at night is really cheap. So they'd get together, 10 of them, and scan each other. Get together literally at midnight. So they would scan each other 10 times each. This is 40% of them. So there's 10 of them. And this is the similarity of brain connectivity between and within individuals. The blocks are within. It doesn't take like a statistician to look at this and say, the greatest amount of variation is between people. See, these are different tasks you're doing in the magnet. That variation is very subtle. It's maybe systematic, but it's super subtle. The biggest amount of variation is person to person. But when we do brain imaging studies, historically, we wash that out. We force everybody to look a little bit more alike. We reduce the on diagonal block, that similarity, and we up the off diagonal. That's what our studies are doing historically. This has prompted within the field a huge rethinking of how we do our studies. There's really these 2 paths, right? We can keep doing our consortia-based studies with thousands of brains. They're expensive, by the way, and it's very difficult to harmonize data across different sites. Maybe you can understand something about population-level tendencies. Or we can start doing more focused studies. Individual brains, lots and lots of data per brain, okay? And that's what I want to talk about next. This is the last bit of the talk. I'm sorry if I go over by just a couple minutes. I think this is actually really profound to see. So the question is, can we use this precision approach in the clinic? Can we use it to actually help people? Some of the early signs from this budding new field of precision functional mapping. There's a study from Chuck Lynch, who's at Weill Cornell. He's studying individuals who have major depressive disorder, okay? Really depressed people, basically. And this is people who have been scanned many, many times. These maps are super reliable, unlike some of the old studies, but it's not 10,000 brains, okay? The hallmark feature that they observe is that a very specific subnetwork, something that we call the salience network, is expanded in individuals who have this major depressive disorder. They don't know why, they don't know the mechanism, but it's reliably so. They see it in their discovery sample and then in 3 replication cohorts. Okay, remember this image, remember this fact. Individuals who have really bad depression, they tend to have an expanded salience network. In parallel, there's a clinical trial happening at UMN and Mayo Clinic right now. It's been ongoing. They're using neurostimulation to address depression. They stimulate parts of the brain. The idea is that somehow it's going to reduce depressive symptoms, help people. They've been doing it for a long time, and they choose the targets where to stimulate, usually on a basis of those group average brains, right? It's not personalized at all, okay? I'm gonna show you an example from one individual. It's an N of 1 study, a true case study. Major depressive disorder, comorbid with PTSD. He's made 3 attempts on his life already, 2 of which he would have died from had people not intervened directly. He's had 19 failed antidepressants that don't work anymore. There's no effect. He's had electroconvulsive therapy 3 times, no effect. This is a person who's profoundly ill, okay? They do longitudinal precision mapping of him, hours of imaging of just his brain. So a lot of energy dedicated to one person. But his salience network is him at the top, wildly expanded. So it's consistent with his previous findings, other precision mapping finding, which is really nice. And they make observations about if it's expanded, it's a zero-sum game. So where has it encroached? What other subnetwork has it encroached on? And those are going to become the targets for this individual's personalized neurostimulation. That is, that we can abate those in some way. By targeting these areas that have been encroached upon, we can hopefully reduce depressive symptoms. It's all done in a personalized way. It is not group average. It's all of the maps are generated for this individual's brain, okay? So you implant 4 electrode paddles. I'm just showing you where the paddles are located. They're targeting very distinct networks, but all with the goal of kind of converging onto this area where the salience network has expanded. Okay, and they do this in a personalized way. There's going to be a whole series of testing. We're going to do it the first day right after it's implanted surgically. They're going to have some parameter optimization afterwards, and then they're just going to let it ride. Okay. So this is what I wanted to show you. Let's see if I can. Hold on. Do this. Do that. Is that better? Good, cool. That's remarkable. Like this doesn't happen, right? And it's large scale imaging. Well, the other kind of cool thing, this is not planned. These are measures of cognitive function, intelligence test, executive function, and then decision making and planning and response speed. Those went up too. Nobody really knows why, but it's all in parallel with this stimulation too. So this is where I end. Again, I apologize if I went a few minutes over. So the outlook for network neuroscience, I think there's a lot to be like wildly excited about. The new nanoscale connectomics is giving us network maps with literally unprecedented spatial resolution. In parallel at the large scale, precision mapping, dense sampling of individuals, is offering new insight into individual differences, forcing us to reconsider our assumptions in the field and opening up really exciting new opportunities for intervention. There's lots of opportunities also to kind of bridge these scales. Primate and small mammal MRI. So again, I said you can't intervene invasively with people, but you can with other mammals, right? So this is an example where they use something called a DREADS, a chemogenetic perturbation to modify circuit level function and silence populations of neurons. They're doing it in a mouse. So not humans, we have the ability to intervene now and make the kinds of measurements that we typically want to make. My colleague or my collaborator, Eve Lake at Yale, does something called multimodal imaging. She records bold fMRI data optically, while she also records simultaneously wide field calcium imaging data. So these two modalities on very different timescales, measuring very different properties of our brain, are being acquired simultaneously. So it's a very exciting time for the field. I think historically, network neuroscience has drawn a lot of inspiration from network science proper, and maybe even lagged behind a little bit. And consequently, it's been spurred on, I think, by advances in analysis tools. We find a new cool technique that new network scientists come up with, and like, this is great, let's apply it to our brains. But I think that's changing in the sense that we have our own considerations coming from our own field in terms of experimental design and how to process and acquire data, and even convergence in terms of what are the appropriate ways to analyze and appropriate measures for our brain network data. So as I'd say, it's a fine time to test our intuition and theory. It's tons of data, tons of questions, good stuff. And I want to acknowledge everybody who was involved in this. Kaio, Maria Grazia, and Bratislav, who did the fly work with me. And then Damien Fair and Ziad, who, I'm like a bit person in that stimulation study, but like you can see all the other people involved. It's a big list. And then my funding sources. And with that, I'll stop. I hope I didn't go too far over.
Thank you very much. Okay, so let's do some questions here, and then if anyone online has questions, you have to say it out loud. Thanks, Rick.
Thank you.
For the fly larva and the dolphin connectome, you do what that? One of the things I saw is that while each annotation had its own, there were overlapping annotations within a single one.
Yeah.
Sometimes they were in multiple. Did you look at that as to why certain annotations were split across multiple?
We haven't investigated that in great detail. There were other statistical issues with the annotations that we kind of focused on. One of them being the annotations themselves, in some cases, are nested. So addressing that in like a statistically rigorous way has proven kind of that was our focus with the annotations, honestly. But I don't know why they bridge, other than I don't have a good answer for that at this point. I think
it seems like the bridging also occurs down the nestedness, so it's like
I
thought it would clarify.
So you're basically saying as you get finer and finer communities, do you tend to see greater and greater specialization, and as you
when you get finer annotation, some annotations are still split.
Yeah.
Or seem to be the same amount.
Yeah, it's interesting. We have not looked at that in great detail. I think it would be well worth our time to invest in that. Yeah.
What's this brain smash?
It's a really cool tool. It's from Josh Burt, and he's actually just moved to John Murray. So it takes a distributed cloud of points. Each of those points has some value associated with it. And they measure something called a spatial variogram, which is a measure of variance as a function of distance. And they measure that, and I don't know the exact generative mechanism, but it takes those data and generates a null value for each point that preserves approximately the spatial variogram. So it tries to match essentially spatial autocorrelation of your data. So it's not quite fair. We know these brains are spatially embedded. Annotations and connectivity are co-localized. So if you were to randomly permute something, you destroy that spatial structure. So it's not really the appropriate null model. But brain smash, and there's other flavors of this too for like contiguous surfaces, but they allow you to try and preserve some of those spatial details, which is really important. Otherwise, it's a way too liberal of a null model with like an unconstrained permutation. Yeah, it's very cool.
Yes, I was wondering if along the that differentiation could be like a scale effect to measure that.
So we're not, I mean, yeah, I wonder about that too. So we have the connectome data are just one snapshot in time too, right? It's like we don't know anything about how it's. So you have one fly, it was doing stuff before, and you don't have any history of that, and so we don't know how it's changing synaptic weights. We don't know how it's just one snapshot. Yeah.
There's some particular relationship with the campus measured there.
Yeah.
Activity is going enough. And in pair, for example, if you have a dimer.
Yeah.
I would say a measure.
Yes. There probably is some relationship. I think the part of the issue that we see with like the large scale imaging, where we might actually be able to measure something across time, is that the temporal resolution is still so poor that if there's, and the spatial resolution is similarly so poor, that changes in synaptic weights, it's just not, it's a very coarse measurement. I don't think
it's very slow.
Maybe I don't know. It may also just be the case. Yeah, I think the fMRI data is still, even if it's there, it's still a very core. What the actual signal it's measuring is still like fundamentally a hemodynamic response. I don't know if it's sensitive enough to capture something like that. Honestly, yeah. If you did like some electrophysiology or something invasive, maybe you could do it. I think in humans. Yeah. Thank you.
It's a broad question, but that last one weights.
Yes.
Do you give a sense of in the community what conversations and ethics for those kind of things looks like? Is there consensus?
So there's a really nice paper that came out it's now 7 years old, but it was on kind of philosophical considerations, not necessarily for this specific type of study, but for brain stimulation in general. So there's people who want to use it to enhance cognitive ability, which would then only be available to people who can pay for it. There's people who want to use it to clinically. There are certain populations that historically have avoided physicians for good reason, and so this type of treatment would maybe not be available to those populations. I am not as in tune, honestly, to the contemporary discussion around this. There are a lot of thoughtful neuroscientists who I think are probably doing a much. I can do some digging and point you towards something, but it's on people's minds. I don't know that there are. It's still so bleeding edge new. I don't know that the societal ethical implications. It's can we do this? Yes. Now let's consider those bigger, broader implications. I think it's still marveling at what we can do for.
The general availability of it?
Poor right now. It's a clinical trial right now. I know that they have done more than one brain now. So it's individuals who have enrolled in this trial, but it's not. I mean, you can do brain stimulation, but not necessarily invasive. And the personalized mapping requires hours of data for an individual brain. So it means coming back to see your physician or see your clinician or see a researcher multiple times. So I think it's still a very select set of individuals who might have access to this. Yeah.
Other questions? Online looks good too. So
cool.
Thank you, Rick.
Yeah, thank you. There's about the chat. Oh, it's people asking about the echo. Okay, cool.